Hi, ladies. Everybody's joining in. One, two, three, 4, 5, 6. A few more people we're waiting on. So if you notice, I've muted everybody today just to start us off. And hopefully that will help our streaming a little bit. The other thing I'm going to do is open up the chat. So in your menu, if you want to open up your chat feature, so that as we're going today, you can type in a question for me as we go, okay? Let's see here. I'm just looking to see who's in, who we're still waiting for. We got Miss Katie and Victoria. And Kirsten is here. I see Hannah and Allison. And Sarah and Keisha. Actually, I think that's all of us for right now. Okay. So if you got my message from earlier today, one of the things that I've learned about Zoom this week is that we're limited in how long we'll be able to meet, how long our meetings can be. They limit us to a 40-minute meeting, which is not really enough time for what I had hoped to do. So I set us up today to have 2 40-minute meetings, one at 4 o'clock and then another one starting at 5 o'clock. We'll see how it goes. I'm going to have to rethink a little bit of how we're going to do these labs, but we'll try to do today's lab the way I had planned, and we'll see how it goes. Okay. Before I get started, does anybody have any questions about anything? I saw that a lot of you have gone through the video lecture, the first video lecture about metabolism. If anybody has any questions or problems or anything with that, you can go ahead and send me a message over chat. And otherwise, I'm just going to go ahead and jump in. Today, we're going to be talking about fermentation. And if you have your lab manual with you, you can turn it to page 117, 117 for fermentation. Today and over the next couple of weeks, as we work through basically the rest of the new lab material, we're going to be talking about metabolic testing that we can do on microbial cultures to help us identify microbes. So there's a whole series of microbial tests that we can, or metabolic tests that we can run on an unknown culture. And depending on the combination of the results we get, it helps us to identify what kind of species, what kind of genus, and what kind of species we have. We've already talked a little bit about this. We talked about selective and differential media. We saw that, for example, we can isolate and select Staphylococcus using things like mannitol salt agar. So we have that available to us. And then starting today, we'll look at some tests that are very specific for metabolic processes. We've actually already looked at one of these. We looked at the catalase test. we look to see whether or not microbes have this enzyme called catalase, which allows them to break down hydrogen peroxide. And today we're going to be looking at a series of tests about fermentation. So there's some great background information in your manual about fermentation. And I summarized a lot of it into some slides that I'm going to show you now. I'm going to try to show you now anyway. Okay, so up on the screen, hopefully you can see the first slide about fermentation. All right, bear with me. So when we talk about fermentation in the laboratory like this, we're really talking about any metabolic process that produces either alcohol or acid or gases or any combination of those inside the culture. And when we think about why an organism would ferment, the answer is it really varies. For some organisms, fermentation is a secondary metabolic pathway. It's not what they would prefer to be doing, but under certain environmental conditions, they really don't have any other choice. So there are a lot of organisms that use cellular respiration as their first metabolic pathway, their primary metabolic pathway. But under certain conditions, they can't do cellular respiration because that final molecule that they need, either oxygen or some other molecule, just isn't available to them. So we haven't talked about respiration in detail yet, but at the end of this long series of reactions called cellular respiration, there has to be a molecule there to accept all of the electrons that have been released from glucose or some other sugar. And for aerobic molecules, that's just oxygen, or sorry, aerobic organisms, that's just oxygen. There are a lot of microbes that do cellular respiration anaerobically, though. They don't use oxygen, they use some other molecule as that final electron acceptor molecule. And for some of these organisms, that might be nitrate, that might be sulfate, that might be a molecule called carbonate. You're going to notice if you look at the formula for those types of molecules that all of them have oxygen in them. So even if an organism doesn't use the actual oxygen gas at the end of respiration, it's going to use an alternative molecule that has oxygen in it. And that's because oxygen is really good at accepting electrons. So when we look at fermentation, an organism might be fermenting because it can't do cellular respiration. It doesn't have its final electron acceptor molecule available. It's in an environment where there is no oxygen, or there is no nitrate, or there is no sulfate, and it's stuck. So it ferments. Fermentation for those organisms is a secondary metabolic pathway. Now, for a whole other group of microbes, they like fermentation. They'll do fermentation just as often as they will do respiration. They do both processes equally well. And then there's a third group of microbes who only ferment. They are what we call pure fermenters. So they use fermentation as their primary metabolic pathway. So it's really remarkable the diversity of metabolism that you see in microbes. These microbes are fermenting as a way to produce energy. And they fall into any of those, that array of classes that I mentioned. So some organisms are doing cellular respiration with oxygen at the end. That's what we do as humans. We are aerobes. So when we do cellular respiration, we use oxygen at the end of that long series of processes. Some microbes are aerobic, so they do the exact same thing we do. But other organisms are doing cellular respiration without oxygen, which seems strange to us, but they are anaerobic, and they're using one of those alternative molecules like nitrate or sulfate or carbonate at the end of this long series of reactions. So it's really important for us to remember that the terms anaerobic cellular respiration and fermentation are not the same thing. It's tempting for us to think that, oh, it's an either or. Either it's cellular respiration, aerobic with oxygen, or it's fermentation without oxygen. But that's just not true. There's a lot of microbes that don't use oxygen, as you know. They don't need oxygen. They don't care about oxygen. They're using other molecules, and they can also ferment. So let's think for a little bit about what's going on. If you remember what you know about cellular respiration, you know it all starts with a sugar. It all starts with something like glucose. You and I use glucose when we do cellular respiration. And we go through this long series of reactions to break that glucose down. The first part of those reactions is glycolysis. And that's when we break that sugar, like glucose, into a molecule called pyruvate. Now, that process, that glycolysis process, doesn't require oxygen. That's not why we require oxygen as aerobes. The oxygen comes in later. So in all organisms, in all microbes, glycolysis doesn't require oxygen. So during fermentation, that first set of reactions still occurs. That glycolysis type of reaction still occurs even though we're in fermentation. So whether it's cellular respiration or fermentation, that sugar is being broken down or oxidized into this molecule called pyruvate. Now, along the way, as a molecule like glucose becomes pyruvate, a little bit of ATP gets made. This isn't the best way to make ATP. This is an inefficient way to make ATP. This is that process called substrate level phosphorylation. And there was a little bit about this in today's lecture. You saw this drawing in today's lecture of an enzyme, sort of a generic enzyme, taking a substrate molecule and turning it into product. And along the way, ATP gets made. The ADP kind of piggybacks onto the enzyme reaction and becomes ATP in the process. This is what we call substrate level phosphorylation. The ADP is piggybacking on the substrate for that enzyme and getting phosphorylated and becoming ATP. So during glycolysis, the first part of respiration and the first part of fermentation, we're making a little bit of ATP. Now, if the organism were going to go through cellular respiration, the pyruvate would continue down this long pathway of metabolism. It would go through the things that like pyruvate processing, and it would go into the Krebs cycle, and it would go down the electron transport chain, long series of reactions. But during fermentation, the pyruvate takes a different path. The pyruvate goes on to one of several products. It's turned into or broken down into one of several products, either alcohol or acid or gas or some combination of those. Now, depending on the microbe and depending on what kind of enzymes it has inside of it, those products can really vary. There are, in other words, there are different types of fermentation. The other thing to know is that glucose is not the only sugar that microbes can ferment. And you already knew this. If you remember back to our special media lab when we looked at that EMB agar, the eosin methylene blue agar. If you recall, that's the one where E. coli gets selected, and we differentiate among the types of fermentation that E. coli can do. If the colonies grew green, we knew that the E. coli was fermenting lactose. And if the colonies grew purple, we knew that the E. coli was fermenting sucrose. So there are lots of different pathways of fermentation and lots of different sugars that can get fermented. So glucose is certainly a very common one. Lactose is also very common. Sucrose is on the list. Fructose is on the list. Here's mannitol. Remember in mannitol salt agar, the salt is the thing that selects out for Staphylococcus to grow. And then if that Staphylococcus ferments mannitol, the agar turned yellow. So there's lots of different sugars that microbes can ferment, and we take advantage of that in that we know which organisms like which sugars, and we can test for those and identify them based on the results. So how is it that one organism can ferment glucose and make an alcohol, and another one can take glucose and ferment it and make an acid? Well, the answer is it all depends on the enzymes that they have, because different microbes are going to use different enzymes as they ferment that sugar, as they break apart that sugar. And depending on which enzymes they're using, they're going to get different products. But the product list is still the same. It's either an alcohol or an acid and or a gas, some combination of alcohol, acid, and gas. So I want us to know the details of two kinds of fermentation, because these are very common fermentation pathways. Lots of organisms are capable of one of these kinds of fermentation. And those two are lactic acid fermentation and ethanol fermentation. So in lactic acid fermentation, an organism is taking that glucose molecule and again, making pyruvate. That's the first step. And then it's going to take that pyruvate and it's going to convert it into lactic acid. You and I are capable of lactic acid fermentation, aren't we? You and I, our bodies, our cells prefer to do cellular respiration. And we do our cellular respiration with oxygen. We're aerobic. But when you get into a bind, and usually that means you are exerting yourself more than usual. Think about, for example, a person who is sprinting down the street to catch a bus. If you get into a situation where you're not getting enough oxygen for respiration, you will temporarily ferment. you will temporarily do lactic acid fermentation, at least until you're able to get enough breath and get enough oxygen again to do respiration. And as you know, when you do fermentation as a human being and lactic acid builds up, it's not very comfortable, is it? It makes our muscles sort of ache and burn when it happens. We would much rather be doing cellular respiration than fermentation. So there are lots of organisms that can do lactic acid fermentation and lots that can do ethanol fermentation. In ethanol, the glucose molecule is first converted to pyruvate, and then the pyruvate goes through a two-step process. It first becomes a molecule called acetaldehyde, and then the acetaldehyde becomes ethanol. And you probably know that we take advantage of this commercially. We brew alcohol through fermentation processes. So all of our local breweries, for example, are growing cultures and encouraging those cultures to ferment so that they make alcohol for us, ethanol. It just so happens that most of the time they're using a yeast microbe to do that, and not a bacteria. So two very common forms of fermentation. But boy, there are a whole lot of different fermentation pathways with our microbes. I've got a few of the products listed here that are commercially interesting. We do fermentation commercially in order to make ethanol. Again, that's the alcohol we like to drink. But we also make butanol. We make an alcohol called butanediol. Those are both industrial alcohols. We make lots of acids. I should say the microbes make lots of acids that we then make use of. Lactic acid, butyric acid, formic acid, succinic acid, propionic acid. All of those acids the microbes are making through fermentation. And for the most common gases that get produced, it's typically either carbon dioxide or hydrogen gas. If you go into your textbook when you have a moment, if you go into our OpenStax textbook, there's a great table in there. This is just a picture of it. It's table 8.3, and it's just some of the common fermentation pathways that we see in microbes and what products get made. They also, I like the table because they also show us what organism is typically cultured in order to produce these products. And then what commercially available things are sold because of these fermentation products. So, for example, lactic acid that is made by all kinds of microbes, including certain species of Streptococcus, certain species of Lactobacillus. That lactic acid gets used to make all kinds of really delicious food, right? Sauerkraut and yogurts and cheeses. We take a lot of advantage of microbial fermentation. All right. So in your lab manual, if you go to, again, that front page, it lists a couple of the tests that we do when we're testing microbes for their fermentation abilities. And we're going to look at these tests today. The first one is called the Carbohydrate Fermentation Test. And this is a test that's going to allow us to see which sugars these microbes are capable of fermenting. The second test is called an MRVP test. This is actually two tests in one. And that abbreviation stands for Methyl Red Voges Proskauer. So MRVP testing. And that test is going to allow us to look for what kinds of acidity are in these fermentation products. And then the last one is called the citrate test. And the citrate test lets us know whether or not our microbes can ferment a molecule called citrate, which is not a sugar, but is another product that gets made during cellular respiration. So let's take a look at these tests. Let's start with the carbohydrate fermentation test. This one's really quite easy to do. What you're looking at here, this is what a carbohydrate fermentation test looks like when you get it from the vendor, when you get it from the supplier. It's a glass test tube, and it has a broth in it, a nutrient broth, and that broth has a reddish color to it because there's an indicator dye in there. We're going to talk a lot about indicator dyes with these tests because that's how we read the tests. We look for color changes. So this is just a general nutrient broth that we add a particular sugar to. Like you might add glucose to it, or you might add lactose to it. You choose which sugar you want to offer to the microbe to see if it can ferment it. The indicator dye that's inside carbohydrate fermentation test is called Phenol Red. And Phenol Red looks red to begin with. So it has a reddish color in the tube normally. And then if the microbe that we put in the tube can ferment our sugar and produce an acid, remember that's one of the products, if the microbe produces an acid, the media will turn yellow. So on this slide, you're looking at some fermentation tubes that have been inoculated and then incubated. And now we're looking at the results. Now, one of the things that is included in the test tube is this little tiny piece of glass in here that I think you could just see. Inside the glass test tube, there's a second test tube, and it's really, really small. It's really thin. It's called a Durham tube. Durham, like the town, like the city. And what we do is we put that Durham tube into the big test tube upside down. So as that tube incubates with the microbe in it, if that microbe is producing any gas through the fermentation process, it will collect inside the Durham tube as a little gas bubble. Now, if you look at this slide, this tube over here on the left that's labeled A, you can just see the Durham tube leaning up against the side. But all that's in the Durham tube is this yellow medium. Now, in the middle tube, you can also see the Durham tube, but look, there's a little gas bubble at the top of the Durham tube. The gas has displaced the yellow medium. And then, of course, over here, this is what a fermentation tube looks like when no fermentation has taken place. So this is what it would look like if we put an organism in there that can't ferment and can't make any gas and is just sitting in there and growing. So on the left-hand side, we would interpret this as an organism that ferments our sugar because the media turned yellow, but it doesn't make any gas. So it's made an acid that turned the media yellow, but it hasn't made a gas. In the middle tube, our microbe has fermented the sugar because we have acid, it's turned yellow, but this time it has made some gas because there's a little gas bubble in our Durham tube. Now I want you to notice something. Whenever we run this carbohydrate fermentation test, we put the bacteria into the tubes, we put the tubes into the incubator. We always put a tube into the incubator that has not been inoculated with anything. We just take a tube, an uninoculated tube, and we also put it in the incubator, and we use it as a control. Okay, so 24 hours later, we're going to pull our tubes out and we're going to read them. We're looking for a color change. Did it turn yellow? That means that acid has been produced through fermentation. And is there a gas bubble in the top of the Durham tube? Now, on this slide, we're looking at some actual results. And I chose this slide particularly because the colors look strange, don't they? Now we're not seeing red and yellow. Now we're seeing orange and yellow. Depending upon the organism that you're trying to test, you can play around with carbohydrate fermentation tubes a little bit. You can put other nutrients into them, and the color of the phenol red can change. This is why you always use an uninoculated tube as a control. So you have something to make a comparison to. So in this picture down here, these tubes were inoculated with 3 different organisms, with E. coli, with an organism called Shigella somnii, and our old friend Pseudomonas aeruginosa. Now, if you look, we have an uninoculated tube over here on the left. So this is what the media started off at in terms of the color. This orangey color, not a deep red this time. And that's because they just put a couple extra nutrients into this media to support these particular organisms. And we're starting with a different color. Now, over here, as you move to the right, this is how the E. coli came out of the incubator. This is how the Shigella came out. And this is how the Pseudomonas came out. Now, we're always comparing our results to our control tube. So the first thing you notice is that the Pseudomonas didn't do anything. It grew because the media is cloudy. The broth has become cloudy or turbid, but it didn't ferment the sugar in here. Now, this particular sugar in this test was glucose. Again, a very common sugar for microbes to ferment. And Pseudomonas is not able to ferment glucose. But look at the Shigella. The Shigella tube turned yellow. That tells us that the Shigella has fermented the glucose, making an acid and turning the medium yellow. At the top of the Durham tube, if you look carefully, there's no gas in there. So Shigella ferments glucose without producing any gas. Finally, here's our E. coli. E. coli is kind of famous for being able to ferment a whole lot of different things. And sure enough, E. coli can ferment glucose. It produces an acid and turns the media yellow. And if you look up, there's a gas bubble right at the top of the Durham tube. You can see the Durham tube looks white up here or clear. And that's because there's a little bubble of gas trapped up there. So we have 3 very different results in this particular carbohydrate test. All right, I have a series of other carbohydrate fermentation tubes for us to look at. And I've got the other tests as well, the MRVP and the citrate. So what I'm going to suggest is we end for now, we close out for now, take a little break, and come on back and join into the 5 o'clock meeting, okay? And we'll go through the rest of the tubes. Does that sound good? Can I see if that sounds good? All right. All right, I'm going to say goodbye for now, and we'll see you at 5.
Sounds good. Sounds good. I didn't mean sounds good. I meant sounds good.